Hi, I'm Dan Heckman, and now we're going to discuss immunology. Now, this is historically lightly tested on step 1, so my goal will be to just provide you with the big picture. Also, there will be some pretty helpful mnemonics that'll help you retain the most important cytokines and cell markers. So without further ado, let's begin by discussing the anatomy of the immune system.
So first of all, what is the lymphatic system? I mean, we'll start talking about lymph nodes, but what is lymph? I mean, as a med student, I was always confused about the exact function, really until I took the time to look it up. You might be the same as I was too, so I'll make it easier for you, and I'll tell you right now. I'll give you the DL on lymph. So, what is it? So, we have these capillaries, right? Capillary, like blood vessels, where normally nutrients and some fluid leaks out too into the interstitial tissue. So where does that fluid go? Well, guess what? That fluid actually becomes lymph. That's right. This fluid drains into lymphatic vessels that then transport this extra fluid back into the vascular system at another location. But before it can make its way back to the vascular system, it has to pass through these little checkpoints, these little balls, these little nodes. And that's why they're called lymph nodes, because this is where immune cells hang out and they filter this filtrate of fluid and make sure that there aren't any pathogens or anything that can harm our bodies. So that is the point of lymph. It's a very simple and basic drawing there, but hopefully that helps you understand what it does and what it is. So let's now get into the lymph node. And like I said, this is the hangout place of immune cells. So here's a picture of one, a nice schematic. Okay, so let's talk about how lymph flows from the outside of these checkpoints or lymph nodes. And let's also talk about the different types of cells that it encounters as it flows through the lymph nodes. So the first thing it encounters is a follicle, and the outer cortex of the lymph node. And you're going to have to know what types of cells are associated with follicles. Do you know? B cells. B cells are associated with follicles. And B cells, they transform into plasma cells, and then what do they make? Antibodies, right? Antibodies go and attack pathogens. Very good. Okay, we'll talk more about them later. So lymph keeps on flowing, and it makes it a little bit farther to the paracortex, which is halfway between the cortex and the medulla, or the inner part of the lymph node. And so in the paracortex are, what types of cells are these? T cells. T cells, we'll talk about them, but there are several different types, helper T cells and cytotoxic T cells. They release a bunch of cytokines, and we'll talk about them later on. So, lymph keeps on flowing. Eventually it makes it to the medulla, and here's where it starts to get really filtered. These macrophages, which are derived from, do you know what type of cell turns into macrophages? Monocytes. So these macrophages filter the lymph as it flows by and try to grab any pathogens that may be present. So speaking of monocytes, another type of cell that is derived from monocytes actually lives in other types of tissue like the skin and outside of the lymph. What types of cells are those? What type of monocyte goes to the skin? Dendritic cells or Langerhans cells. And they're called dendritic because dendrite means branches or roots. And so they have these little root-like webbed feet. And these are extra good, as you can imagine, at grabbing pathogens. So they bring pathogens and fragments of pathogens from the skin or wherever they're coming from. In general, they're in contact with the outside environment. So they bring pathogens from the outside environment to the nearest lymph node and present them to other immune cells to stimulate an immune response. So that's the purpose of a dendritic cell. And from there, the lymph flows out of efferent vessels and it back into the bloodstream eventually.
So a way to remember that efferent vessels exit a structure is just that. It starts with an E. And a way to remember that afferent vessels arrive at a structure is that both start with an A.
One important note is that blood can actually filter into lymph nodes as well, and it can do it. Do you know where it does it? It does it in actually the paracortex, so in the T cell domain. And specifically, what's the name of those vessels where blood filters in? They filter in high endothelial venules. So I think of it as high because the blood's saying hi to the lymph node, so it's filtering in. That's how I remember how it's coming into the lymph node. Kind of silly, but it works. Okay, so looking at our entire body, how does lymph flow? So we could go through every single area of your body and talk about what node it drains to, but in general, just remember that whatever artery is nearby is whatever lymph node that lymph will drain to. So, for example, lymph around the stomach that's supplied by the celiac artery, that's going to drain to, you guessed it, the celiac node. And lymph around the groin area is going to drain to inguinal nodes because that's the artery that supplies it. So just remember the arteries and you'll be fine. And in general too, lymph keeps draining from one node to the next until it travels all the way up to the thoracic duct, which drains almost all of the lymph in the entire body, except, what's the exception? The right arm and the right half of the head drains to the right subclavian vein. Alright, but everything else goes into the thoracic duct. Alright, now it's time to talk about the spleen. So, there's a lot of analogies between the spleen and lymph nodes. So, the lymph nodes, what are they responsible for? For filtering lymph. Well, the spleen is responsible for filtering blood, okay? So that's important to keep in mind. But there are follicles in the spleen, just like lymph nodes, and they're the same types of cells, B cells, T cells, and macrophages, but they're just in different locations. So just like lymph nodes have their special locations for special types of cells, so does the spleen. And we'll talk about those in a little bit. So the spleen is located in the left upper quadrant. That's important to know because it's a common site in trauma of injury, of blunt trauma to the abdomen. It's very vascular and people can actually bleed out from their spleen. So the spleen is broken up into sinusoids. What are sinusoids? They're little chambers with different components to them that we see here. We have, first of all, surrounding the spleen is a fibrous capsule. Just like the lymph nodes had a fibrous capsule, so does the spleen. And second of all, so I said the spleen is responsible for filtering blood. So blood flows in via arteries, right? And it leaves via veins. That makes sense. So when blood trickles in, the first thing it encounters is this white area here. What's this white area called? White pulp. Makes sense. Looks like pulp and it's white, so that's why they named it that. And what cell type is present in this white pulp? I'll give you a hint. There's abundance of follicles. So what's in follicles? B cells, right? So just like the lymph nodes, where we had follicles in what part of the lymph node? The cortex. We have follicles and B cells in the spleen. So I didn't mention this before, but there are two main types of follicles. The first is primary follicles, and those are solid in color. And those are a bunch of B cells that have not been activated. They have not been stimulated by antigens yet. And the second type of follicle has a pale center, and those are secondary follicles, and those have been activated. Those are B cells that are actively proliferating and responding to antigen stimulation. And again, what do B cells make? What do they synthesize? Antibodies, right? What's this type of antibody that has 5 parts to it? It's a pentamer. We'll talk about antibodies later, but this is Ig immunoglobulin M. Okay? That's the first type of antibody that B cells make before they class switch. And the spleen is actually very, very important in synthesizing IgM. And IgM, in turn, is very important in attacking bacteria, especially encapsulated bacteria, bacteria with capsules. We'll talk about that in a little bit. So that's important. It's a cool little animation there, right? Woo! So, blood has trickled into the white pulp, and now it's spreading even farther. What surrounds the white pulp? There's these things called PALs. Periarterial lymphatic sheaths that surround the white pulp and the arteries. What cells live there? T cells. T cells live in pals. Okay? T cells are your buddies. They're your pals. They help everybody. Helper T cells are your pals. That's how you remember that. And that's where they live, and they secrete cytokines and do all the things that we'll talk about in a little bit. the normal T cell functions. Okay, so we talked about the white pulp. What's this red substance here on the outside where blood eventually reaches? Well, it's red in color, so it's going to be red pulp. And what cell lives there? Macrophages. You knew they had to be somewhere, so here's where they are, in the red pulp. And they do just what they did in the lymph node, where they reach out with their little arms, their little pseudopods, and grab onto pathogens. Filter the blood through that, through mechanical filtration. So that's the spleen, and that's the sinusoids of the spleen, okay? So we're talking about encapsulated bacteria, right? And we were talking about how IgM is important in destroying encapsulated bacteria. For reasons we'll get into a little bit later, but actually, do you know what pathway is activated by IgM and IgG? It's the complement cascade, and that's actually important in lysing a hole inside of this capsule of the bacteria. And let's discuss then the different types of bacteria that have capsules. So there's a helpful mnemonic called SHINE skis, okay? And this helps you remember all the different types of encapsulated bacteria. So the S stands for strep pneumonia, H is for Haemophilus influenza, N is for Neisseria meningitidis, E is for E. coli, the other S is for Salmonella, K is for Klebsiella, and the last S is for Streptococcus agalactiae. Alright, so those are all the encapsulated bacteria. That's just a nice little microbiology review. So, what happens then if this person were to experience trauma and their spleen were lacerated and they lose their spleen? Well, then they won't make as much IgM and they won't be able to combat all these encapsulated bacteria. That becomes a major problem. So this person is at risk for sepsis, and they're going to definitely need to get vaccinated against especially Streptococcus pneumonia, probably Neisseria meningitidis too, so they don't get sepsis by those bacteria. So, another random topic that you might be asked on boards is, if they do lose their spleen, then you'll see these special types of cells in their blood. What are these? They're red blood cells with little dots in them. What are these dots called? They're called Howell-Jolly bodies. And normally, the spleen, the macrophages in what part of pulp? What pulp? In the red pulp of the spleen, filters these little fragments out of red blood cells. But without a spleen, there's no filtration. They remain in the red blood cells, and they're called Howell-Jolly bodies, a sign of splenic dysfunction, okay? It's important to know. Now you know why. So now it's time to talk about the final lymphatic organ, and that is the thymus. So we'll talk about this in more detail later, but in general right now, just know that just like the lymph nodes, the thymus also consists of a cortex and medulla, okay? Which is very similar to lymph nodes. It regresses with age. Starts out enormous in a kid, right here in the anterior mediastinum, and then gets smaller with age. So what's the point of it? What's the purpose? It's a site of T cell maturation. And T cells, what they do is they start out in the cortex, okay? Start out here, and they make their way into the middle, into the medulla. You can think of the medulla is where the T cells mature, okay? Both start with M. So that's what you need to know, and we'll talk about that in a little bit. Also know that the thymus has special cells. What are these called? Not really significant in any way, but they are called Hassel's corpuscles. Ooh, fancy. Swirled calcifications. Not to be confused with Samoma bodies, but these are called Hassel's corpuscles, and they are unique to the thymus. Random embryology tie-in, where is the thymus derived from? It's a good question, right? So, it is actually from the epithelium of the third brachial pouches. Review your embryology, if you didn't know that.
Alright, time for a flash quiz. What types of bacteria are targeted by the spleen? So we just went over this, and they are encapsulated bacteria, and those include all of those SHINE skis organisms. Strep pneumonia, Haemophilus influenza, Neisseria meningitidis, E. coli, Salmonella, Klebsiella pneumoniae, and Streptococcus agalactiae.
Very, very good. You got it. Lymphatic structures down.